First of all, I'd like to thank to the Professor Proik. So you're here. So I actually for your devotion to organize a symposium in spite of the corona situation and many difficulties. So thank you very much, and also thank you very much for inviting me to the symposium for introduce my recent research. So this time I want to introduce drug discovery with the FMO method. Especially I want to introduce our method construction and also some of our experience using the FMO calculation. Okay. Usually I don't make this kind of animation, but we start the company. So company, they pay for this animation. So I make this one. And so actually there are many good tools developed and now in the frontiers of structure-based drug discovery. So we're trying to construct our drug discovery platform with these new technologies. So, okay. Actually, we want to introduce a new computing system as quantum computing, and also we usually introduce the energy calculation for the protein with the quantum mechanical method, even protein ligand docking, and also we want to see whether our design structure is correct or not. So we introduced the cryo-EM. We bought two cryo-EM system and now it's running. So now we construct this system. Still, quantum computer is not, actually it's early stage of introducing to real application. Maybe in 3 years or 4 years, then we can run quantum computer for solve our system. So as you know that for the drug discovery, there are two important things. First one is how can we accurately calculate the energy and structure. Another one is how can we mimic the natural process. It means how can we make an algorithm for mimic the natural process. In the case of energy and structure calculation method, only a few people working on that. So even my student, they don't like working on this one because it takes a long time and less paper and takes a long time for the graduation. So some more people are working on this one, but still it's much less than the people who really working on drug design. In that case, many people are working on that. For energy and structure calculation and this algorithm development, maybe they majoring the physics and physical chemistry and computer science and so on. So, but it's very hard for most of people for studying this field. Instead, they want to develop some drug and design drugs. So many people working on this field, and they use mainly commercialized software. Now the commercialized software is very nice for using. So even this situation, still we need to calculate energy and structure very accurately. It's very important. So during the last 50 years, maybe more than 50 years, many of the famous and good scientists are working on developing force field still is very useful and much progress. The accuracy is, became better and better, but still not enough for the drug design. So even as we start developing a force field since 1987, we finished 2 years ago. Even though we put a lot of energy for developing this one, but no one in my laboratory used this force field. It has some benefit, but not much difference with the other packages. So there are limitations of the pairwise force field. Where it comes, it mainly comes from the multi-body effect. With the two-body pair interaction description, we cannot describe that kind of a multi-body effect. So that's a limitation. And recently, neural potential is very powerful. So actually, they doing a deep learning with the big data. It's a potential surface calculated with the quantum mechanical way. So many big data, they training non-linearly, and then they almost produce the energy calculated with the quantum mechanical calculation. So now it's a very strong but some shortcoming, but still, it became very strong. And also, we can use the quantum mechanical calculation. So there are 3 ways to use the empirical method and the quantum mechanical calculation or neural potential. Now neural potential works very well. So first I want to introduce the neural potential. So actually, Andrian Roitberg, Florida, he developed the ANI model, any models. So they training with the potential surface of a huge number of molecules, and then they calculate, compare. So with the DFT calculation, it takes like 52 hours. I don't know which molecule they trying, but maybe some packages of molecules. But AN1 model, it takes only 0.3 seconds with CPU. So RMS deviation is only 1.0 kilocalorie per mole. It means that it's very accurate, but it's very quick. So, and none of the empirical force field produce this kind of accurate correlation. So I think it's a very powerful tool in ELV in the future. So here, they developed the neural model for each atom type. So they don't discriminate like SP2, SP1, SP3, so they don't. Only atom type. Atom is like a carbon, hydrogen, something like that. So they calculate the atomic energy based on the environment. So if you put the environment of the atom, central atom, then you can get the energy of the atom. So once you get this model, then you transfer to the molecular calculation. So in the case of H2O, this is the model for the oxygen, this hydrogen and hydrogen. So when they calculate energy based on their environment, and then you can get atomic energy. If you sum those atomic energy, then you can get the total energy of the system. It's very powerful. So when they prepare the potential surface, they can be used for the training. They carefully choose the potential surface. They use the molecular dynamics and the normal model analysis and some torsional space sampling. Then they get the large number of the DFT calculation. Then they can get the model. And also they select the subsampling. That is more important, much contribution conformation than they do coupled cluster calculation. They get a more accurate model. So usually they develop anyone models. This is the first version. In that case, they can calculate only CHON models. So it's quite limited for the calculation. Now they announce they expand to the sulfur and halogen. So if we combine together, maybe they don't have phosphorus, but we can calculate most of the organic compound for the drug design, and also protein. So they have 4 models they proposed. So we want to check whether they developed this model for the single molecular energy calculation or conformation, but we believe that it may work because atomic environment is not different, whether it's chemical bonded or it's a non-bonded interaction. So we tried whether we can use that for the intermolecular interaction. So this is a crystal structure of a 10 complex. Then we trying to reproduce the crystal structure with ANI2 models. So this is a result. This RMS deviation is very, very accurate. It's very accurate. So after we get this result, we think that this ANI models can be used for the protein ligand complex interaction. So it means that we can use that for the drug design. Now we will move to the FMO. So FMO model was originally developed from Japan. So actually they start to develop when I was in Japan. It was, my age is around 30. So a long time ago they start to develop, and recently it's quite popular for using FMO for the protein structure calculation and the binding energy calculation. So actually Professor Morikuma and his student is Kitaura. So they developed together this FMO method. And also there's actually Kenichi Fukui, he get the Nobel Prize with the frontier beta theory. Actually, he was the professor of the Keiichi Morikuma. So there's some kind of a tradition for molecular calculation in Japan. So for the FMO, there are needed 2 models. So first one is the energy decomposition models. So they decompose total energy into the electrostatic exchange, polarization, and transfer, and this. So Kitaura and Morikuma, they develop, and also Ziegler and Rauch, independently they develop the same energy decomposition models. And also another we need is how can we fragment big molecule into the fragment. So they assume that the total energy is the fragment energy plus between fragment interaction energy. So if we have this kind of assumption, we can calculate the energy and structure of big molecule like a protein. This is an example. If we have a protein, we can fragment those protein with the amino acid unit. So this is amino acid unit. This is the distance between the amino acid. So this is a fragment. First, we get the electron density of the fragment with the low level initial calculation. After that, we regard other fragment as a perturbation. So we modify the electron density. So we do that for each of the fragment. Then we can get the modified electron density. And after that, we continue this kind of procedure. So finally, we can reach it to the converge. So we find that this converged wave function, electron density, quite almost exactly same to the wave function of the molecule without fragmentation. So it's a very powerful method. So once we get this kind of calculation, we can get the interaction energy. Interaction energy of the each amino acid contribution and so on, this energy composition. So with the FMO, we construct drug design platform named Avengers. So those procedure is quite similar to the normal virtual screening procedure. So we can get the we can define pharmacophore, but when we define pharmacophore, we introduce the quantum mechanical calculation and especially pair interaction energy terms and also its component together. With that, we analyze the pharmacophore. So when you're doing the screening, virtual screening, we used several kind of energy calculation method. Among them, CDE, those three, the FMO-based method. So we do the energy minimization with the FMO method. In that case, we used some approximation tight binding method, but even though it's approximation method, is the complex structure is quite accurate compared with the experimental data. So this is our system. And so we checked that whether this FMO method can be used for explain the free energy of the binding get from the experiment. So here we can this is the experimental. I should fix the value, and we change. This is the interaction energy obtained from the FMO. So it makes a plot. It's a very good correlation. So after this calculation, we think that we can use the FMO method for explain the experimental. I should fix the value or binding free energy. But there's some error because in the experiment they have a entropy term and the free energy, but FMO calculation is only internal energy, so that's a difference. But without that kind of free energy error, the experimental data and calculation value is very correlated. And also we apply for this method for the PD1, PD1 interaction. So this is the interaction between PD1 and PD1. So we calculate the FMO method, and here is the distance between the amino acid. This is PD1 and PD1, and also the color is the interaction energy. So once we get this one, we can automatically generate this kind of interaction map. So this interaction between PD1 and the PD1. So, and also we do the same thing with the PD1 and PD1 antibody and PD1 and PD1 antibody. So we mapping that, which is the most important amino acid contributing to the protein-protein interaction. So it means that we must block those amino acid will be a good PPI interaction inhibitor. And also we apply that for the COVID. So actually in China, they isolate the spike protein and purify, and they get the cryo-EM structure, and also they find the glycoprotein inside, and then they put to the PDB. So after we got the PDB, we got the structure from PDB, we doing the FMO calculation. Then in 2 months, we published and submit. And so in 2 days, it's in BioRxiv. So recently, many people use this information for develop COVID and human AC2 interaction inhibitor. So we get the information, we give the information, and there's a difference, some interaction between COVID-2 and AC, and COVID-1 and AC. There's some difference, but many of the interaction is quite well correlated. But some interaction is different from COVID-1 and COVID-2. So those are pharmaceuticals for the COVID-1. So we can, if we make a plot together, then we can see that which kind of drug for the COVID-1 can be used for the COVID-2. And also we apply for the this one for the YEPTAD PPI interaction inhibitors. It's a HIPPO pathway.
2 minutes left.
5 minutes left, right?
2 minutes.
My time is 30 minutes.
Yeah, sure, okay. Yeah.
So we apply for the HIPPO pathway here. So when HIPPO pathway is very important, so it's control the growth of the cell or organ and so on. So it's a tumor suppression. But if it's dysregulated, it moves to the cancer. So you, in normal case, there's a phosphorylation of the YAP, so it inhibits the transcription and control the size of the cell or organ. But if there's a deregulation, there's many transcription, then the organ became very big size, and also fly became very big size. So it's a good target for the anti-cancer drug discovery. So this protein has 3 interfaces and 1 central pocket. So after we analyze the interaction with the FMO, interface 1 is not good for the drug discovery target because the many interaction, but it's a very weak. So interface 2 is a very strong interaction. It's a very simple, so it's good for the drug discovery. Interface 3, there's a very strong interaction, but the pocket is very wide. So we decide to inhibitor for interface 2 and interface 3. Interface 2 and interface 3, and doing same way, analyze and FMO calculation, and we get good inhibitors and also central pocket. So we get the good result, and for interface 2, we licensing out this compound to the pharmaceutical company, and we're still working on this one. And also central pocket, we get the sub-nanomolar compound. So now we are ready to move to the animal test. And also we find this DNA binding pocket. So we also find out the inhibitor for this DNA binding site. So inhibition is very good for the, because this is a DNA repair process. But for anti-cancer, we must inhibit this binding. So we're also working on this one. And also we apply for that the protein engineering platform. So here we use the phylogenetic tree and cryo-EM structure, and also protein binding analysis with the FMO, and also phenotypic perturbation and neural potential and so on. So I show you one example. So this is the So the superoxide dismutase. So for FMO calculation, we get which part is very important. And also we do the friendly perturbation. And then we find 12 candidate for the mutation, only 12. So after mutate experimentally, we test this activity. So this wild type is the commercially used wild type. Compared with this is the half-life. It's very, very small. It's a minute. So we can get better activity, and also the half-life is almost twice. So it means that it's a very powerful FMO method for the protein engineering. So I introduced briefly quantum computing because in near future it will be a very important tool for the drug discovery. So this is from the McKinsey report. They said that quantum computing can be a game changer, especially for the industry. First industry to benefit will be a pharmaceutical company or a chemical industry. Now the big pharmaceutical company, they think that this technology can be a strategic stance. So maybe a promising in the future. So many of the pharmaceutical companies in the United States and Europe, they introduced the quantum computing and paid a lot of money. And also there are many startups, they developed the algorithm, quantum algorithm for the drug discovery component. And also there's a consortium, and also maybe around 10 quantum computer providers. The strongest is IBM now. Problem is that the quantum computer, because quantum computer use the superposition of the state, but the problem is the quantum computer, there's uncertainty principle in the state. So there's many errors. So classical computer, the error is very, very small, but in quantum computer, the error is very big. So it means that the order is 24 orders in magnitude and in the error. So the problem of the quantum computer is how can we reduce error? So how can we stabilize? So quantum volume is how can we calculate the big size of the job? So now in quantum computer, we are here. It's a 2020. It's like incubation period. In 10 years, we must reduce the error. So error correction is very important. But this is our research centers project. First one is a generative model development. Second one is molecular dynamics. Third one is quantum computing. So after that, we can use the quantum computer for the drug discovery freely, but very powerful. And now the IBM is the most powerful. Maybe in next year, they will announce maybe a 10 qubit. 10 qubit is maybe the computing power itself is almost infinite compared with classical computer. So now here, but in the horizon one, is quite near, but in the horizon one, actually we can apply the quantum computer for drug discovery and protein structure prediction, quantum chemistry, and material design. So actually we published the quantum computer chemical problem with the quantum computer. Is quite simple, but we want to check that whether this quantum computer can be used for the real chemical calculation. We calculate essential reaction, so it exactly same with the accurately calculated with the classical computer. So we make this kind of consortium in Korea. So in summary, FMO is a very useful tool for calculating electron density and energy of protein. Using FMO, we can calculate the docking study and analyze the docking mode and the energy decomposition. Is very useful for that. And also neural potential will be very, very important quite soon. And also we test the Avengers and the carpet for experimental case, and then we can reproduce very well, and also we design very useful enzyme with those methods. Okay, so here usually two groups. This is company, this is my research center. Usually method developed from my research center, and here they use the method, and also they develop drug, cryo-EM, and the medicinal chemistry and biology. Okay, thank you for your attention.
Okay, thank you, Professor Taino. Unfortunately, we don't have any time for a question. You can have a look in the chat. There is a question from Igor Polakov, and reply them there if possible.